for joining. This is, I think, our first joint seminar that we've organized between the Close-Knit Network and the Neuromod Network. So welcome to members of both networks. Just to let you know, I mean, the rationale for doing this as a seminar was that, as many of you know, we had a meeting between the networks held in Nottingham earlier this year. And at that meeting, people who had been funded in I guess, the first funding rounds from both Close-Knit and Neuromod gave talks to update the networks as to what they'd been doing. And I think it was very successful, but unfortunately, several people were unable to make the meeting at Nottingham, and we wanted to give those people a chance to also present to members of the network. So we organized this as a special seminar series. It was a special one-off seminar event, I think. By the way, I'm Andy Jackson, for those of you that don't know me, and I'm involved in the Close-Knit Network. And so it's my pleasure to introduce our first speaker today, who was funded in our first funding round of the Close-Knit Network. And that's Weiko Yoshida, who's going to be talking about controlling social interactions using real-time fMRI neurofeedback. And Weiko is a research fellow at the University of Oxford, and I'll hand it over to you. So I'm very much looking forward to hearing your talk.
Okay, thank you very much. Can you hear me? Okay, good. So thank you for giving me the opportunity to talk about my project, which is supported by Close-Knit. I'm a computational cognitive neuroscientist working in Oxford, and I'm Japanese. And I moved to Oxford, 3-4 years ago, and so I've been interested in like higher or decision making in human behavior, and especially computational modeling of behavior and imaging data. And then also I'm interested in neurofeedback as a new technology to understand the basic science, basic brain mechanisms, and also as a potential treatment or support for a patient. So this aim of this project is to develop a new novel type of MRI neurofeedback system, incorporating social learning context and computational behavioral modeling. So, okay, good. So there are several types of neurofeedback, but I'm focusing on functional MRI neurofeedback, which is a technique that allows participants or individuals to train and regulate or modulate their brain activity in real time by providing feedback based on their brain signals. And this kind of closed loop processes helps individuals to learn to control or modulate specific brain regions to enhance their cognitive or emotional or behavioral outcomes. So this is a basic standard system. Like we get imaging data and process it, and then feedback to the participant. But this technique is used to understand brain mechanisms and also used to support several patients. But yet there is no kind of established standard method. And then there are a variety of a way to conduct this experiment. So typically, so for example, as a feedback, most of studies use a visual feedback, how good or like how good their brain activity in terms of it was close to the target or not. For example, we sometimes give like an actual number, like a 15 or 20 is better and 30 is better, and the participant try to increase the number, or also we use the height of the bar on the size of our discs, and the bigger is better, and so on. And also some studies use auditory feedback, like high tone is better and low tone is not good, and things. But during the neurofeedback, the most of the studies, what participant does is look at a blank, image or do nothing. And then experimenters asked them to control their brain activity, get a better feedback. And then I have done several this type of standard type of neurofeedback experiment, but subjects is really confused. And then it's a huge effort to control their brain activity because they don't have any information from environment, so they can do anything. And actually, after the interview, after the experiment, we interview them, and most of people say try to think about good things or counting or try to imagine like a learning, his learning and so on. But several things, it's always quite random. And then because controlling state space in a brain activity is massive. So it's really hard to explore efficiently, we feel like. So therefore we thought maybe, and then also this type of like a freedom in a neurofeedback, during neurofeedback is quite difficult for patient or elderly people, younger children, because they get bored and they lose attention and they get tired of things. And so we came up with an idea. Maybe we can ask the participant to perform the task at the same time of neurofeedback experiment to try to let them engage into the neurofeedback more. And then another thing is another part of the neurofeedback system is how to make a target brain activity. The one is like how to pin down the region of interest, so how to localize the area. And then one way is if you have a very clear hypothesis or knowledge about which brain area is correlated with what you want to see, you can use anatomical localizer. But most of studies use a functional localizer. So basically they do like a very simple task beforehand, the neurofeedback in the same subject, and then look at the brain activity and then pin down which area they use in the neurofeedback. But so this is working, this works fine for if you are interested in like a quite simple mechanisms like motion related area or visual information or memory. So you can use like, for example, memory task. You can perform like a very short n-back memory task before and pin down the memory related area and so on. But as we are interested in like higher order function as the social learning, we try to use a computational modeling to pin down this target brain area beforehand. Okay, so it's on talks. Okay, so the aim of the study is I summarize here is we try to implement a novel neurofeedback system that designed to explore kind of higher order cognitive computational, higher order cognitive functions in social context. And as I said, by integrating task-embedded neurofeedback approach and computational ROIs, we aim to investigate, we hope the neurofeedback effect could be enhanced. And and also we conduct a pilot experiment to examine whether individual can use neurofeedback to control like kind of higher order brain activities, and also this brain activity change their behavioral performance. And then additionally, we also seek to explore the clinical utility, potential utility of this system. So to do that, we had like a PBI meetings with autism community and then try to understand what the issues and what the future direction should be. Okay. Okay. So so this is one example what I have done before, but I think I will skip it because I don't have time, but in this experiment, instead of traditional neurofeedback, like, I don't know if you can see its cursor or not, but the traditional feedback is just a decoder give you the feedback based on brain activity. But here we use the reinforced learning controller, like basically an agent learn to control their output based on the brain activity. So basically, this is a dual adaptive neurofeedback with system we conducted. So I just wanted to show that we have done something before, and then this is a kind of more, you know, the better version of previous experiment and things. So okay, so here we focusing on social learning because I'm quite interested in like like people's social interaction and things. And social learning is a key process for acquiring knowledge and behaviors based on social interaction. And then one important mechanism is called observation learning. So observation learning includes a choice imitation, which is just replicating or mimicking others' action. And then the more important thing, I think, is global emulation, which means based on their action, we try to understand others' intention, or let's say value or goals. So this effect of observation learning was first demonstrated in Bandura's Bobo doll experiment in 1961, which showed the children not only mimicking the aggressive behaviors they observed in others. So basically, this boy observed an adult playing with a doll before, and then experimenter let him to play with it. And then he showed very aggressive behavior because he observed it. But interestingly, children not only mimicked behaviors they observed, but also even expand to them using their imagination to take the behavior further, such as, so he tried to shoot the doll with a toy gun, etc. So this experiment concluded that children model behaviors rather than just imitating them. And then this observational learning is impaired in autism spectrum disorders, ASD, and due to a typical activity in the brain regions like amygdala neural area or those medial prefrontal cortex. And so we believe like a recent neuroscience technologies include neurofeedback could be a potential technology to support their behavior. So based on those background, we designed a new experiment to embed into the neurofeedback system. So in this observational learning experiment or task, we use two players perform the two armed bandit task individually first, and then after that jointly. So basically this task have two phases, individual learning task and observational learning task. An individual learning task is a simple two armed bandit task, and then the participant selected one of two stimuli, like two fractal images on the display by pressing the corresponding button. That's it. And then after making a choice, the participant received the outcome point as a number between 0 to 100. So 100 is better and the 0 is not good. So they repeat this process and try to learn the goodness or value of each fractal image. And then after that, they do the observational learning task. So here, two players perform the same task, but without by observing each other, but not outcome itself. So in this observational learning task, there are two phases, and one is action phase and one is observation phase. And action phase is a very simple, the same as individual learning task, and participants choose one fractal images out of two, but they do not observe the outcome itself. They do not get the point. They can just, choose again and again. And then during the observation phase, participant observed other players' response. So basically, you are observing other players performing the same task, and then you can see others' choice, what they have chosen, but you cannot see the outcome. So, and the two players repeat those two phases many times, repeatedly, and update their own values by observing others' behavior. And then how do we set up the task is experimental setting here. So, so we use here like 5 fractal images in total, but in individual learning task, we use only 3 cues per subject. And then, so yeah, so the outcome was drawn from the Gaussian function with a mean is, for example, here it's like 13, 50, 70 with a standard deviation 12. So for example, for subject A, this yellow one is about 50, and then like a funny, well, the pink circular one is about 30. And those 3 stimulus, they learn the value based on trial by error. But the opponent or another participant or agent learned a different set of like 3 cues during their individual learning phases. So this 1730 is overlapped. They both learned individual learning, but that is these 2 images in yellow one and this one, which gives outcome average 50, is like a kind of, they learned as separately. So they have a kind of, they learned private values, privately learned. And then in the observational learning task, we use the 5 cues presented in total include, so 4 cues which we use in an individual learning task, plus 1 mystery cue, which none of them learned before. So for a subject, there are 2 novel cues, 1 mystery cue completely unknown, and 1 cue learned only by the opponent. So by observing others, you can get a kind of sense of which is better and which is not good. And then we did like 3 sessions with different fractal images, and then each session contains 90 trials of individual learning task and 200 trials of observational learning task. I don't know how long we have, but okay. So to try to understand their behavior, try to explain their behavior, and also try to find neural correlates related to this observational learning experiment, we introduced a computational model based on the TD learning, which is a reinforcement learning method, and DDM, drift diffusion model. So I don't know if I have time to explain, but so TD learning is a reinforcement learning method where an agent learned the value based on the prediction error based on so the actual outcome and the predicted outcome. So here in this figure, which means the delta is the difference over their choice itself and then estimated value. And then in standard TD learning, so this upper part of this figure, the learning rate of this how to update the value is constant, but more realistic approach suggests it is influenced by stimulus-dependent associability or reliability. It means for some cues, you are quite confident about it, so you don't need to update more. But some cues you are not sure about it, so self-confidence level was low. You should update the value of that based on observed actions. So we called it as like a self-confidence level, and estimated from like a prediction accuracy in the individual learning task. And then they also, in a social context, it's crucial to assess the reliability of others' behaviors. It means if you are not sure if the others are confident about their choice itself, you should not use their behavior to update your value. If you cannot trust their behavior, you should not update that much your own value. So this others confidence level, estimated others confidence level, we make a model to estimate the level based on their response time. For example, if they choose if they made a choice very quickly, we believe they are quite confident. But if one person did not spend so long time to choose one, I feel like he's not confident, so I should not update the value based on that. So we introduced two quite new dynamic learning rate into this conventional TD learning model. And then we found our model fit better than other models without those confidence level regulations. And then, so to look at the neural correlates, we looked at the brain activity correlated with these three components that describes how much you update your value based on observation. The green one is actual prediction error, and alpha self-confidence, and kappa is other confidence. And we looked at brain activity and then found those three quite strongly related parameters is actually like represented in the brain, a kind of overlapped area in inferior lateral prefrontal cortex. And then we found it's quite interesting. And actually this area is a part of, major part of like a social brain. And also it is known this reduced this IFG, inferior frontal gyrus areas activity, or reduced connectivity with this area, impaired understanding of others' action. And then emotion, and then also learning abilities in a social context. So it's kind of our finding is consistent with previous findings. So we decided to use this area as a neurofeedback target area. So here we aim to embed this observation learning task into the neurofeedback system. So here we use the agent as a reinforcement learning agent who learn the value of cues based on participant action. So basically, in the observation phase, the participant observing agent behavior. So agent is a learned, performed the individual learning task before as well. So they know they learn the values of three stimulus. So that's kind of informative. Their agent behavior is kind of informative, relatively informative. And then in action phase, participant choose action, and the agent is actually observing the participant action and update their value. So your actual action modulate agent behavior as well. For neurofeedback, what we have done is we recorded the brain activity during the observation phase, and then based on this brain signals, we gave the feedback in the action phase. So here we introduced a new thing, like a 3 stars showing in the middle of the display. But basically, your brain activity was good. I mean, like inferior frontal gyrus activity was higher than baseline, you get a 3 stars. So basically, this participant aimed to regulate their brain activity simultaneously enhancing their performance, observation learning performance by observing others' behavior. So at the same time, they need to do like a behavioral task and also a neurofeedback task at the same time. And this is experimental design, so it's a quickly explained, but as a control subject, we have two groups. One is upregulate group and one is downregulation group. So one is trying to increase activity, one group try to decrease activity. And, and the before and after that neurofeedback experiment, we asked them to perform the two cognitive tasks. One is a social inference task. This is it's quite a similar task, but a two-armed bandit task with social information. And then basically in this task, the participants make a choice. And after that, they are given the choices of other four performers, which is we actually collected from the actual subject. And then they can decide if they change their choice or confidence level. So we can see how much they take into account others' information into their second choice.
So
And then
Wake,
also,
sorry to
yes.
interrupt, but can I just say we've got about 2 minutes left so that we don't
Okay.
run into the. Thank you.
2 minutes. Okay, I'll finish in 2 minutes. And then another one is a simple no-go task to ensure that there's influence on the inflammatory control. And this is experimental system. So we have like two control systems, and then one is a processing imaging, and this is online neurofeedback calculation and a task experimental computer and things. Okay. So we have quite a lot of technical issues, to be honest, in that system. And after resolving those and adjusting the parameters, we managed to scan 2 participants in this project. We don't have a concrete result to show yet, but the system is working well, and we are now focusing on collecting more data. And this is just an example result of ROI activity of so basically inferior frontal gyrus during the task. The gray areas observation stage and red areas action stage. And then, so this is actual data. And then we were hoping that activity is going up in observation phase compared to action stage, or also if the observation stage activity go up during neurofeedback. But from those 2 subjects, I could not find that type of significant effect yet. But so we are trying to make it more based on this data. Maybe we can change that, use a different type of baseline and so on. We are still featuring the parameters as well. Okay, so also we have done some PBI activities because we explore the potential application for this system in our autism patient, and then we presented our task to like ASD people, and then also working with some to improve the task. But interesting is the outcomes was basically they feel like their difference from normal people is like kind of it's just diversity, and they don't want to be treated, so they don't feel like they want to change, and they are, and also they feel like learning from like by other people with autism does help a lot. They feel like they know how to do that. They know how to learn social information and things, but the way of learning is different from other people. That's what they say. And so it's quite interesting, I thought, because we try to change them. That's really, I felt it's quite an arrogant thing. So not like trying to change their brain to environment. We should try to change the environment to fit their way to do. So this project is supported by CrossNet and also BRC in Oxford. And then here's my students and then some research assistants. Thank you very much. Sorry.
Thank you very much, Wego, for an excellent talk. Really interesting study and very interesting data. So I'm aware of time. We started sort of 5 minutes in, but I want to make sure we leave space for our other networks.
Yes.
If anyone has any quick questions. We've got one in the chat, and also I'm sure Waco will be able to answer other questions through the chat. But let's do this one. So we've got a question here. I don't know if you can read it, but maybe I'll just read it out. What's your approach for minimizing the time delay of visual feedback you give to your participants, as in the stars? And do you think that alternating the observation and action trials in some way might disrupt the neurofeedback? Yeah, I had a sort of similar question. Are people able to learn based on feedback that's given quite some time after the epoch that the brain activity is measured in?
Yeah, that's quite a big problem in actually MRI feedback, I think. That's why the EEG is quite commonly used. But having said that, it's processing itself takes only about less than 1 second to pre-process. So we can actually give like almost immediate feedback, like a continuously, for example. But I thought it's more like a distracting because it's changing like every second, and it's really hard to control or like online. So I thought offline system might be better, but of course we don't know how long we should take. Now we are taking five trials about 10 seconds, 14 seconds, but I don't know if it's good or not because it's fluctuate within 10 seconds. So those kind of things we need to explore a bit. But
yeah. Well,
yes.
a challenging project, but actually really impressive progress that you've made, and very, very nice data. So what I'm going to suggest is that if people have further questions for Waco, please put them in the Q&A, and I think Waco should be able to answer that through the Q&A. But thank you very much for your talk, and I'm now going to hand over to Simon, who's going to introduce our next speaker. Thank you.
Thank you.
Thanks, Andy. So I'm actually here with my undergraduate tutorial students who are here to sort of experience some research, sort of real research presentations, possibly for the first time. I'm not sure. So for those who don't know me, I'm Simon Schultz. I'm the PI of the other of these networks, the NeuroMod Plus network. And it falls to me the pleasure to introduce Sophie Morse. So Sophie Morse is an Imperial College Research Fellow and a UK Dementia Research Institute emerging leader in the Department of Bioengineering at Imperial. In fact, her lab is sort of just near mine. So we have plenty of interactions. And I'll just, without further ado, hand it over to Sophie to give a presentation on ultrasound modulation of neurons and glia. So thank you
so much. Thank you. Can everyone see my mouse?
I can.
Okay, cool. Okay, so, oops, sorry. Very sorry. That is just typical that my cat decides to make an appearance just as I'm about to start. Okay, so basically I'm going to talk a bit about what my lab does in terms of using ultrasound as a therapeutic tool to modulate the activity of neurons and glia with a specific focus on glia. And I'll then give context as to how the project that was funded by the NeuroMod network collaborative grant that was in collaboration with Simon Schultz and James Choi at Imperial, how that ties in to creating a device to help us understand the mechanism behind what's happening with ultrasound in this context. So when we're using ultrasound as a way to basically image the inside of the body, which is what we normally know ultrasound for, we're talking about relatively high frequencies above sort of 1-2 megahertz. But when we're using ultrasound for a therapeutic purpose, it's quite different, both in terms of the frequency range that we're talking about. So we're more in the range of hundreds of kilohertz up to about 1 or 2 megahertz. But what's also different about the way ultrasound is used in this context is that instead of having an ultrasound transducer emitting waves in front of the transducer straight ahead, the waves are focused. So essentially you have an emitter emitting these waves and having a either geometric focus or an array of transducers that will focus that energy onto a specific point. So what that means is that you can have these transducers on the outside of the body, right, non-invasively stimulating areas that are inside of the body at a specific target without affecting the tissue that is between that emitter and that focus. Now, this technology, focused ultrasound for therapeutic purposes, is being used for all sorts of different applications. And one of these applications is for neuromodulation, right? One of the most exciting ways of using this is, can we stimulate and suppress the activity of neurons in the brain? And the great thing about this technology is not only it's non-invasive and it's targeted, if one wants it to be, in the sense that it can be more or less targeted, and you can also scan the entire brain if you need to, but it's also that it can generate reversible effects if one wants to again, and nothing happens between that emitter and that focus, with that focus being able to reach any area of the brain. So depth is not an issue. Now, using ultrasound in this context, researchers have discovered that by pulsing the ultrasound in different ways, you can actually generate stimulatory or suppressive effects. One of the obvious parameters to do this that has been found is actually the pulse repetition frequency. So how often you fire those pulses actually is more likely to generate a stimulatory effect if it's faster, and if it's slow, it's more likely to generate a suppressive effect. So these kind of changes in parameters, not only repetition frequency, but also pulse length and how long you're modulating for, will have an influence as to whether you can generate stimulatory or suppressive effects. Now, the mechanism related to this kind of treatment, which has been investigated for all sorts of clinical applications, including OCD, depression, and schizophrenia, for example, the exact mechanism is unknown, but what we do know is that mechanical and thermal effects can be involved here, both in terms of mechanical effects, in terms of actually generating changes in the mechanically sensitive ion channels that sit in the cell membrane, but also thermal effects if certain parameters are used where long enough pulses or high enough intensities are reached to actually generate a build-up in energy. Now, my group is specifically interested in using this technology not just to modulate neurons, but also to modulate the glial cells of our brain, and in particular being able to modulate the brain's immune response, both in terms of the inflammatory levels, but also the actual innate immune cells of the brain, as well as in other contexts beyond sort of neurodegenerative diseases, which is where this is most exciting in terms of applications for also being able to modulate the peripheral immune cells that maybe enter the brain in contexts such as for brain tumours or any diseases where the blood-brain barrier is leaky. Now, in this context, we're particularly interested in the glial cells, which include cells like the microglia, the astrocytes, the oligodendrocytes, and all of these cells in the brain have so many different functions. And we're really sort of talking about immune responses, surveillance, we've got synaptic support, phagocytosis and clearance, and all of these cells are not only involved in all these different functions, but they're also really attractive therapeutic targets because their functions are altered in so many different disease processes. So the idea here is if we could use focused ultrasound as a method, therapeutic tool, to modulate some of these functions that are being altered in disease processes, then this could potentially help us to either delay progression of a disease or potentially reverse some of the effects that have sort of chronically built up, for example, a increase in inflammation in the brain, for example. So with that in mind, a lot of what my group has been doing over the past year or so has been sort of setting up both in vitro and in vivo systems to be able to study the effects that ultrasound has on these glial cells. And one of the cells that we're really interested in and have sort of started off with, although we're expanding to oligodendrocytes and astrocytes as well, is microglia. Now, microglia are the innate immune cells of the brain. And as you can see from this image, they're involved in all sorts of different functions, all the way from immune functions, so they release pro and anti-inflammatory cytokines to signal to other cells that there's an issue in the brain and that they need other cells to be recruited, or tell other cells to do specific things in terms of downstream signaling pathways. We've also got phagocytosis roles that are really important with these cells. They clear out unwanted substances, protein aggregates that build up. They're involved in pruning synapses, which is really important in development, but also many diseases. This is where things sort of start to go, well, not start to, but go wrong. And support functions, both in terms of supporting oligodendrocytes, astrocytes, neurons, etc. Now, in disorders, various diseases such as neurodegenerative diseases, for example, these microglial functions start to be altered in various ways. Throughout the disease process, there'll be various things that change. And some of these processes include chronic inflammation building up, specifically sort of in later stages of the disorder, reduced phagocytosis. This can actually happen quite early on, and so the sort of build up of protein aggregates, but also loss of that support function, and so we start to see sort of neuronal damage, etc., which is not just because the microglia are altered in terms of function, but that's one of the processes that's involved here. So I was telling you that we've sort of started setting up in vitro and in vivo models to study this. And one of the first in vitro models that we used was actually in collaboration with Andrea Caffarelli and Leonardo Ricotti at the Sant'Anna Institute in Pisa. These collaborators have this sort of system where in a water tank you can basically have 3 different ultrasound transducers. They can also be exactly the same. And you have these 3 slots, 4 cells, and you basically have these cells grown in a sort of insert, and you just insert it into this slot. And basically the top and bottom is just a very, very thin membrane, which essentially prevents ultrasound reflections from building up, standing waves from building up, so that your cells are essentially receiving the energy that you think they're receiving, rather than it building up and it being more than you expect. We've also got an EMRA student in my group who's now a research assistant, who's built a system for our own lab here at Imperial. Very simple system, and here doesn't even require an insert. The cells can be grown on a film, and it all slots into a device that's very easy to use and simple, because that was kind of the idea. Now, the first experiments that we did started off very simply with just having microglial cells in, just growing them, inducing an inflammatory state in these cells with the LPS, which is a bacteria that creates inflammation, and then looking basically after a day whether the ultrasound stimulation of these inflamed cells at various different frequencies, pressures, and durations led to a difference in the levels of pro and anti-inflammatory cytokines. Also with qPCR, we've now done gene expression levels, differences in gene expression levels of these pro and anti-inflammatory genes, and looking at that 24 hours later after the ultrasound. We've also done some cytotoxicity analysis as well. Now, these first results, and remember there's limitations here, right? It's just it's a system with just microglial cells. We do not have the complexity of the brain, which is what we're going to look at more in the context of the in vivo experiments. But this is trying to set up a system where we can just initially have a look at maybe giving us a bit of direction in terms of parameters to look at more closely in vivo, but also in vitro setups can be particularly useful for looking at mechanisms. So that's why we sort of set this up to start with. And one of the first things that we saw in this very simple system was that ultrasound, basically, let's say these cells are just by themselves in a control setting, they release a certain amount of pro-inflammatory cytokines like IL-1 beta and TNF alpha. If you induce inflammation, that shoots up. And then when you apply ultrasound in those cells that are inflamed, essentially these levels are coming down across a range of different pressures and a range of different durations, even with durations as short as sort of 5-minute treatments. And with TNF alpha, so the results are stronger with IL-1 beta. With TNF alpha, it seems to be sort of trend maybe towards a reduction, but this was 0.2 megapascal was the only result that seemed to be a bit more significant, and also in line with the IL-1 beta. And so we took this parameter set with 5-minute stimulation. This is a center frequency of 500 kilohertz, so something that's relevant clinically. And when we looked at the gene expression levels of pro-inflammatory genes, we see that this reduction is sustained for at least a sort of period of 72 hours within these this sort of in vitro system, whilst anti-inflammatory genes over time, over this 72-hour period, are increasing, or there's a tendency toward this increase across those 72 hours. So essentially here we basically got some initial data to show that with ultrasound we can reduce inflammation in these microglial cells across a variety of different parameters, and that inflammation can be sustained for at least 72 hours, and something to look at further in terms of the duration of that and how long it would be sustained for, essentially. But in parallel, what we've been doing is looking in vivo at stimulating with ultrasound, with focused ultrasound, the brain of healthy mice to start with, because we weren't really sure start what we could modulate, what it is that we can increase or decrease, and how much control we have over the technology. And so we wanted to explore this in, of course, a sort of an initial system, just a healthy mouse, looking 24 hours later. Here we're treating the whole brain, and we're looking, basically we're keeping different parts of the brain for histochemical staining, and also looking at protein and gene expression changes in the future. We started off by using, in this in vivo study, 3 different pressures, 3 different pulse lengths per pressure, and we're talking about 1 megahertz here, so a slightly higher frequency, more suitable to the size of a mouse skull, although we've also got some ongoing experiments in vivo with 500 kilohertz transducer. The main difference here compared to the in vitro study, and this is because this was for a different study in slightly different context, is that here we're treating the whole brain by sort of scanning it, and each area of the brain is only treated for 6 seconds. So it's a really short amount of time that this focused ultrasound is on for. And what we're looking at here is a whole range of different markers for different cell types. We've got microglial cells, we're looking at their morphology, but also clearance and phagocytic activity, neurons and their activity levels, astrocytes in terms of morphology changes. I've got a student now focusing on oligodendrocytes and myelin, looking at MBP, and then we've got both excitatory and inhibitory synaptic markers, and all of these protocols have been optimized and are now working. And we're also in the process of doing ELISA results, which I won't show today. I'll focus mainly on just showing you some results with the microglial cells, which are particularly interesting. So what we're seeing here, and this is an image, sort of zoomed out image of microglial cells and their phagocytic levels. And if we zoom in, we can basically see with IBA1 in red, we have these microglial cells branching out, and these cells are very motile. So when they become more activated and start to change function, they sort of withdraw their processes and can move around a lot easier in a sort of more amoeboid or like a shape. And CD68 is a marker, is a lysosomal marker, and it gives us an indication of how much phagocytosis is going on in these cells. And when we look at those different pressures and different pressures and different pulse lengths, what we see quite clearly is a trend with a higher acoustic pressure towards an increase in phagocytic levels, both in terms of how much each cell is phagocytosing, but also an increase in the number of cells that are phagocytosing. So both of these aspects are increasing with the 2 higher pressures. And this trend seems to be, this is at 1 megahertz, and this trend seems to be constant across different regions of the brain, which is interesting because, of course, the ultrasound focus, the center of the focus is towards the middle of the brain, towards the middle. If the hippocampus was more stretched out here, it's towards the center. And so there will be areas of the brain which are receiving slightly lower pressure than a 1 megapascal. If that's here, then they'll be lower up and down. But we're seeing similar trends across the different areas of the brain. And also, in terms of safety, we've done one of my undergraduate students has done a lot of safety analysis, both in terms of very carefully identifying whether there are any red blood cells extravasating, so a sign of the blood-brain barrier being open, and this is H&E staining, and basically between sham and treated we have no differences at the highest acoustic pressure, 1 megapascal. So we're assuming that at the lower pressures this would also not be the case. But also he started doing analysis for detecting any cell death in terms of apoptotic cell death as well, which are results that hopefully we'll have soon. So from this overview, we're seeing that from this very initial data, we're seeing that ultrasound can modulate phagocytosis, this clearance activity, potentially increase it, but it can also modulate inflammatory signaling pathways and reduce inflammation if cells are in vitro state are sort of in that inflammatory state. So we've got in vivo experiments ongoing where we've created that inflammatory status in the brain, and we're looking at how ultrasound, whether basically the results in vitro correlate in vivo. But along all this path and in future work, what we really want to do is also look at what is the mechanism here, what is going on with these cells, and what we want to do is be able to see both simultaneously and also afterwards during that ultrasound modulation treatment what is happening to the cells and different expressions of levels of ion channels, etc. And so this is where we felt that multiphoton imaging could come in as a really strong tool to investigate this kind of mechanistic aspect of what's happening during these ultrasound treatments. And so with the Neuromod network collaborative grant with Simon Schultz and James Choi, we looked into, we managed to hire a research assistant, Jiho Kim, and he looked into different devices that already existed, and there's sort of a couple of different devices, both with 1 photon imaging, 2 photon imaging, and there's a couple of papers there, all very sort of, there's nothing that you can just buy and use basically, it's all very experimental and built in-house. And so what we set out to do was to create a device that could simultaneously stimulate with ultrasound and do 3 photon imaging to achieve that higher penetration depth in the brain. Now, this is no easy task, especially because building transducers is a very fiddly job and really takes a lot of skill. So I'll show you some initial results from this. Jiho, it was his first time building a lot of these transducers, but our goal here was to work with a 16x objective lens and have basically an array of transducers around that lens that could focus at the same point as the focus of the objective lens, essentially, so that we'd be stimulating an image in the same plane. Frequency of the ultrasound 500 kilohertz. Our goal was to emit 200 kilopascals at least and 100 cycles, and he initially wanted to sort of fit in about 11 transducers in this array shape, but actually given the working distance at the center of the focus of the transducer, he could only fit in 8 because of the dimensions that the ring needed to be to have that distance. So he started by doing a whole load of simulations, both without the objective and then with the objective lens in the simulation. And as you can see with the objective lens, actually, the focus is becoming slightly shorter, which is not necessarily a problem at all. And actually, from these simulations in K-Wave, by calculating the sort of pressure that's at the very center of the focus, you got a 13% increase in the pressure at that focus when the objective lens is present as well. And that's the objective lens here. And that's probably because of reflections of the ultrasound taking place and sort of building up more energy at that focus. But we were pretty pleased with this in terms of the focus and what we could do with it. And so he started building the transducers, which is quite fiddly, but basically involves building a sort of 3D print in a case, calculating how thick your matching layer needs to be based off the wavelength, having a back-end layer so that you're basically dampening any reflections of the ultrasound as the transducer basically vibrates so that it doesn't interfere. And in the end, he ended up having 2 matching layers to reduce the amount of ringing that he was getting of the pulse at the very beginning of the pulse. And that seemed to help to have this double matching layer in place. And these are expertise that James Choi's lab has. And so this is what it looked like, basically the design with the different transducers. They look quite long because that back end layer is necessary to sort of dampen those vibrations. And when he went on to calibrate his transducers that he made, we can see that the pressure field of these different transducers looks a little bit different for each of the elements, especially when you make them by hand and don't have the funding to just buy these transducers. So you can see they're all a bit different, but I can tell you for certain that these look a lot better than the first ones he built, which had pressure fields that looked really quite funky. So this looked relatively good in terms of a first prototype of this kind of system. And when calibrating, so seeing what the pressure field was when having all of these different transducers in place in the array, he basically needed to align the negative peak of the pulses emitted by all of the transducers so that they align at the focus. Basically they reach at the same point. So I had to introduce various delays, and then this is what his final calibration looked like in terms of the focus. So we sort of got dimensions of about just over 5 millimeters in the Z direction and 2.5 and 1.4 in the X and Y. This is what his sort of pulse would look like. So he managed to reduce the dampening, the sort of ringing effects at the beginning by having that double matching layer, which was great. And in the end, this is what the device looks like in terms of these are the transducers. This is the sort of ring array around the objective lens. And unfortunately, we were meant to do our first in vivo experiment a couple of months ago, but the laser of the free photon has been needing fixing and hasn't quite been solved yet, but we're hoping to be able to do that very soon and get some exciting first results. In practice, the system needs quite a lot of improving, both in terms of the fact that currently the output with 6 elements, which were the only transducers that ended up fully working by the end of the project, because these have got sort of epoxy resin, sort of 3D printed casing. And if you drop them, they break really easily. And we sort of get an output of about 53 kilopascal at the focus, while our aim was 200, so potentially with more elements and a better driving system, which would require further funding. We could improve this even further. So it's going to be exciting when we manage to do these first in vivo experiments with this system. And with that, I'd just like to sort of conclude that we're looking at how focused ultrasound can be used as a therapeutic tool to modulate the brain's immune response. And we're in the process of developing this device to be able to understand better the mechanism of this procedure and to simultaneously basically perform ultrasound modulation with free photon imaging on awake mice with Simon Schultz's system, with even behavioral testing while doing these types of treatments in the future. So with that, I'll just conclude by just mentioning Jiho Kim, who did most of the work. He's here, the research assistant, James Troy and Simon Schultz, and Go, who are part of the sort of wider collaborative team, and you for listening. And I'm very happy to take any questions. Thank you.
Thanks, Sophie, and apologies for the laser. It turns out that the lasers for 3-photon microscopes for the aficionados are basically, they're very hard to keep running. The pulses have to be really, really short, sort of 50 to 60 femtoseconds. And as a result, everything becomes very sensitive. So keeping it all running is difficult, but it should be back up soon. So hopefully
I'm looking forward
we'll hear more. Yeah. Do we have any questions? I think people are supposed to put them in the
Yes, so there is.
Yeah, we have one here. From Jake Toth, really interesting talk. Thank you. What do you suspect the mechanism is? That's a big question, but yeah.
Yes. I mean, I think the mechanically sensitive ion channels, there's no question that these are involved, and there have been in vitro experiments and that have looked into that, and they're definitely involved, piezo 1, piezo 2, but also a whole load of other ones. I think thermal effects will come more into play when you're using parameters that allow that build up in energy. So we're talking about maybe really long pulse lengths or high intensities, or maybe even more when you're trying to generate, for example, in neuromodulation treatments, more suppressive activity in neurons, potentially there a thermal effect could play a stronger role, but it's probably a mixture of both depending on the parameters that you're using. But it's a great question that we really want to find out more about, basically.
Thanks, Sophie. I've actually got a similar question, or a related question. So you in the talk that you gave, you were sort of mostly talking about simulating glia. Now, separately, there's an interest in, and there's separate work on stimulating neurons. And I'm just wondering, are there differences, like in terms of the targeting glia versus targeting neurons? How can you know that you're not affecting neurons when you're targeting glia, and if you're targeting neurons, how with pulse parameters for that, how do you know you're not targeting glia as well, not affecting glia as well?
No, I mean, it's a great question, and it's part of what we're looking into. We don't expect to modulate the glia and not affect the neurons. I think that would be quite weird in a way. And a lot of what we're doing now, we're looking at the neuronal activity as well, is actually showing differences there as well. So it's more that when we start to try to understand the mechanism, it'll be interesting to see whether it's a direct or indirect effect and which way it's going, basically, and try and understand that to get a better gist of essentially what parameters may perhaps first influence the glia and then the neurons, or vice versa. And so that will be an interesting.
Sure. Obviously, the 3-photon is one way to answer that, but
yes, exactly.
Heck, we just need to get some electrodes in as well, right?
Yeah. Yeah.
Sure. Any other questions? Yep, there's one from Rufus Mitchell-Heggs. Really great talk. A couple of questions. Eventually, how do you envisage obtaining focused ultrasound feedback in the human brain? Have you considered spatial transcriptomics to observe in vivo immune response? Is there any risk from scattering causing local hotspots? That's 3 separate questions, I guess.
Okay. So I'll try and go. So the spatial transcriptomics, absolutely. If I had more funding, we would have already done it. So
not cheap, actually. Yeah.
Not cheap at all. So yes, that's always in the back of our minds as something we want to do. The scattering, yes, that's why the simulations are particularly important, right? And even in the device, the array device that I was showing that we've built, there is some build-up of energy at a distance sort of equivalent both before and after the focus. So if that ended up being inside of a mouse brain, then that's something we'd need to account for and try and correct for, or essentially try and modify our device to eliminate. But that can happen. But with simulations and careful design and improvements and iterations, prototyping, etc., that should be eliminated to some extent. The other question...
Human brain was the other question.
Yeah, human brain, and it was about feedback. Yes, I mean, a lot of groups are working on this kind of with neuromodulation towards closed loop feedback systems. I mean, Nottingham, for example, and that's something that's in development. So hopefully it'll sort of, by the time this sort of gets to a stage where it could be used clinically, that'll be developed and working really quite nicely. Yeah.
Okay, thank you, Sophie. I think we have run over time now. I'd just like to thank everybody for attending, and thank both Wako and Sophie for some wonderful talks. So thank you all.
Thanks, everyone.
See you next time. Bye.
Bye.